Okay, this is drawing 44, and it's the last drawing that's specifically about T cells. And we're going to see where they are born, what the types are, and how they make decisions for the immune system. So I'm just going to zoom here on the top part, and we're going to draw right here a little picture. I'll show you ahead of time. It's going to look like a torso of somebody. Now, this is very easy to do, and don't panic because it doesn't have to look perfect. All you have to do is draw, find out where the middle is, right about there, right? Move over a little bit from the middle. You can leave a little dot there. Come down like this, over, make a shoulder, and down. And then do the same thing on the other side, like this, and down. And then over here, kind of go up like this, and down. And over here, kind of go up and down. Okay, and it looks cartoony. It's not supposed to look like Hercules or anything, okay? Just real cartoony. Okay, you know what? Right here before we start, you know how we've been drawing every cell and kind of making realistic? Well, if you're wondering, you could just draw a little note right here that looks just like the B cell, a circle within a circle, and then you could put T cells look just like B cells. So just in case you want to make a little note for yourself, staying consistent with all the drawings. These things look just like the B cells when they're small, when they're in their naive state. Can't tell the difference. Okay, we need to draw a little organ right here, right? It'll be kind of right above where your heart is, a little thing like this. It's like a little kind of butterfly or whatever. You see, if you look for pictures of them on the internet, you'll see them look in all kinds of blobby, weird shapes, but that's fine. And we're going to label this Thymus, T-H-Y-M-U-S, Thymus. That's a very obscure organ. You know, everyone's heard of stomach and heart and lungs, and a thymus. If you say to somebody, "Where's your thymus?" they probably don't know. It's right now. Let's make a note here in parentheses, under rib cage. So if you put your hand on your chest, you're not going to feel your thymus, right? It's going to be under the rib cage. Now, another note here, let's put the thymus is large in children and small in adults. This is a very important point. If you go looking for the thymus in an adult, you're going to have a hard time finding it. In children, it's very large. There's a lot of T cell action going on here. And then as you go through your teen years, it shrinks smaller and smaller and smaller. And then as an adult, there's not a whole lot left. It becomes, the cells sort of turn to fat. It's kind of more fat and not so much active, not very much active thymus left. So a lot of the action in this organ goes on when you're a kid. Okay, I'm going to draw a little piece of bone over here. This is the end of your, don't waste any time being artistic. We just need to know, okay, that's the end of your arm bone. And some little cells. Of course, these are way out of scale. They would be microscopic, but let's just pretend there's a bunch of, these are going to be T cells forming in the bone marrow. Okay, so, and we'll draw an arrow here from these cells over to the thymus. Put T cells, and from here. Now, you know, of course, that the T cells aren't going to, this is not the direct path that the T cells take. How are the T cells going to get from the bone to the thymus? Well, there's only one way. What's the highway system in your body? Your bloodstream. So they're going to jump into the bloodstream, and they may circulate around the body quite a few times before they finally get off at the right stop. The T cells have a little shape matching. There's little cells inside the thymus. It's like if the T cells had one side of the Velcro and the thymus cells had the other. Okay, when they roll by, well, some are going to stick. It's a shape matching thing. But these guys have receptors that fit T cells, and as they roll past and they go through there, they're going to stick. And that's how they get there. They migrate. So T cells are, quote, born and the bone marrow, like all leukocytes, then they migrate to the thymus, and that's why they're called T cells. T is for thymus. In the thymus, the T cells will differentiate into either CD4 or CD8 cells. That's what we just saw. They're going to be either CD4s or CD8s when they come out of the thymus. One or the other. And in the thymus also, number 2, they will be tested to see if they will not attack body cells but will attack pathogens. 98% are going to fail the test and they'll just die and be recycled, discarded and recycled. Only 2% are going to make it. And it's a very elaborate system of shape matching. There's cells inside here, the endothelial cells in the thymus express all the different body proteins. And the T cells, they bang around and bump into them. And if they react to the body proteins, bang, they're killed. Okay, because you don't want the T cells attacking the body proteins. And then conversely, there would have to be, who would be testing the T cells to see if they did recognize pathogens? What kind of cell would be in here doing that? Yeah, dendritic cells and macrophages. There's some of those, the dendritic cells that hang out in the thymus, and they're like the professors that give the tests. You know, like some people say you're going to be an engineer. Some engineers are out actually building buildings, but a few engineers have to go to colleges and universities and teach students. And so the professor dendritic cells and macrophages sit in the thymus and help teach the T cells. I don't really teach them, but basically the only T cells that get out of the thymus are the ones that do recognize body cells, first body cells, and do recognize pathogens, and they don't attack the wrong thing. Now, occasionally some things can go wrong. The world is not perfect, and unfortunately, some people have T cells that kind of can get mixed up. But that's the plan. The plan is that they know what they're doing when they leave the thymus. Okay, so that's why they're called T cells. They go mature in the thymus, and then they leave the thymus and they go and they will sit in lymph nodes and in the spleen. A lot of them go to lymph nodes. So let's make a little arrow here. Once they leave, they could go to lymph nodes, more spleen. Some will go to the tonsils, to lymph nodes. I'll put or tonsils. So some of them might migrate up to your tonsils, right? And then you've got lots of lymph nodes under your armpits and your chest and your groin. There's little areas, and your neck has a lot of them too. So they'll go and migrate and sit in the nodes, and they'll kind of wait until they're needed, because a lot of them aren't going to be needed, but you'll need a few. For the lucky ones, they're going to become activated. Okay, I don't know if you call that lucky, then they have to work, right? Okay, so down here, when CD4 and CD8 cells graduate from the thymus training school, they go sit in lymph nodes and tonsils until they are needed. Most will never be needed. So we're going to draw a cartoon version of these guys, all right? So we got to give them a head. This is the... Let's pretend that's the actual cell. And if you want to give them a ring, they have these giant nucleus, or not. This isn't really necessary, but you want to make them look realistic. So the main thing is we're going to put a face on them. We're going to put little eyes that are kind of looking up, and if you just kind of go like that, they look kind of lost. They don't really know who they are, what they're doing. They kind of look innocent, right? And we're going to give them a body. Remember the B's looked like the letter B? Well, this is really easy because the T's, if we give them one that looks like the letter T, right, it kind of looks like they have arms. Isn't that cute? Okay, they have cute little arms. And you know what we could even do, you know, it'd be really cute, is to put those little receptors in there so that it looks like they have little weird hands. How about that? There's their little T cell receptors. It's cute. Yay! Okay, so there's our little T cells, and we're going to put a 0 on their chest. Like in sports, they're number 0 because they're naive. They don't know anything. I know number 0 it doesn't mean that, but here a lot of times if you see T0, 0 mean naive. So here they are, question mark, eyebrows going like that. Who am I? Where am I? I don't know anything. I'm naive. I have no idea. And then they're also often labeled T with a small letter H for helper. So we know but this guy isn't, this is a helper. And this is a naive T. And we could put killer, TK. I like that, but in books they generally go with that cytotoxic word, so they have to put a letter C down there. So he's the helper and he's the cytotoxic killer. But he is the killer. Okay, so we'll put CD8 killer / you have to know cytotoxic. And this one is the CD4 helper. Okay, and they sit in the lymph node. We could put them in a little cute lymph node here. Okay, and this guy though, this guy has something else. When he got to be a CD8, he also got a little weapon. So we're going to put, he's got like a little holster or something on the side here. He's got a little gun, and sorry for politically correct people, but we have to make it a gun. It's a perforin gun because it really does like shoot little bullets into pathogens and make little holes. It really does. So he's got a little gun, but look at him. He's not going to use it. He may never. He doesn't know what to do with it. He doesn't even know he has one yet. So these are called naive cells. Naive doesn't mean stupid. It just means you don't know anything. You don't have any experience. They're capable, but they don't know it yet. Now we have to take a tiny time out here and we have to talk about the bad guys, the pathogens. Okay, pathogens can be what? Viruses, bacteria, protozoans, yeast, worms, okay, anything that doesn't belong in your body like that. Now, some of these guys stay outside your cells and some like to get inside your cells. And so we're going to do this side first and then go back here. So we're going to draw a couple of these guys, and it makes a difference to your cells where you're going to look for them. Now remember, your cells don't have eyes, do they? How do they know where to look for things? They don't have eyes, so they can't see where the pathogens are hiding. Now let's draw some of these guys. Let's draw some body cells. Just kind of blobby. These are average. These aren't supposed to be any particular. Maybe they're epithelial, maybe they're not. I don't know. And then underneath here, let's make it so that this is gonna be our little capillaries, our endothelial cells. Now, this is not an intensive study of pathogens. This is just a little aside, but this makes a difference where they are. And I got this from a medical school professor lecturing to medical students, and he made a big point of talking about this. So I thought, yeah, I think I'll put that in my lecture. Okay, so let's give them some nuclei here. You could even go like this real quickly. Look, endoplasmic reticulum, mitochondria, whatever. Okay, make it look like a little cell. All right, I'll give this guy a nucleus. So remember, the endothelial cells are cells. They look like flat pancakes wrapped in a tube shape, but they're cells. Okay, so there's our capillary going by and our body cells. Okay, so I have a list down here. Pathogens generally stay outside the cell are called extracellular, and the pathogens that like to hide inside are called intracellular. And I have a short list. I just kind of compiled this from the internet, and you don't have to memorize it or anything, but I just think it's kind of interesting. I'm like, ooh, what goes inside cells? I don't know, I think it's interesting. I'll show you pictures of just a few of them. Okay, so the things that like to stay outside, anthrax, which is famous for affecting sheep and cows. And here's a picture of the anthrax. It's a little bacillus. These are the anthrax. So these would just be body cells, animal cells. And these little guys are the pathogens. So that's what anthrax looks like. Famous for affecting livestock. You've probably heard of E. coli. Most E. coli found a lot in your gut. Cholera in the intestines. This is a big problem. I think let's see, we have, I think this is the cholera guy right here. It looks like a sausage with a tail, sort of. Here's this guy. So here's the outside of him. These are other things behind here, but this is the It's called a Vibrio shape bacteria. And what else? Meningococcus. If you've heard something that has meningitis, it affects your brain and your spine. It likes to make you get a lot of pain. It's a very dangerous infection in the spinal cord and the brain. And what Clostridium in your intestines, Borrelia, Lyme. A lot of people have trouble with Lyme disease. And the people in my family have it. I'm very well acquainted with it. A little spirochete. It generally stays outside your cells. It can take different forms, but it's generally considered extracellular. And some strep, you've heard of strep throat and staph. A lot of times that's like on your skin. Different kinds. There's group A, whatever. But strep and staph can be extracellular. And let's see, what else do I have here? Let's go for the intracellular. All viruses, automatically, all viruses, right? Viruses can't really live. If they were to be outside a cell, it just wouldn't do anything. It would sit there. To replicate, a virus has to get inside your cell. So by definition, they're intracellular. Listeria found on food. And I think maybe this is salmonella. Salmonella, this is a picture. You've probably heard of salmonella, knows someone to get food poisoning. And it's also listed as intracellular. This is the best picture I could find. I guess these little things are all the salmonella. And get close enough here. Now, does it look like they're inside? Like the pink things. Of course, this is all stain, right? Real cells you can't see, but a couple of these look like they're inside. But that was the best picture I could find. And let's see, Candida Yeast. Oh, this is a cool one. I'll show you. Yeast has these little round buds, and it has these things. Isn't that weird? This is microscopic, so I don't know, that would be a cell, and you get these yeasty things, but they can also apparently get inside. That's a picture of it sitting outside, but it can also go and hide inside. And I thought that was kind of different. And the last picture I have there is on the list says Yersinia, the bubonic plague. And here's a bunch of Yersinia bacteria. They look like they're little tiny bacillus joined end to end, these long strings of bacillus. And they stain pink, so I guess that would be, I don't know, gram-negative maybe, I don't know. Not sure if it's gram-positive or negative. But that would be the bubonic plague bacteria. And then Mycobacterium TB, tuberculosis. And malaria inside. That's the last picture I have. Malaria likes to get inside your red cells. So these things are erythrocytes, and there's the little protozoans. It's not a bacteria. It's a protozoan, like a paramecium is a protozoan, amoeba. It's a little single-celled thing. There's one inside there. So malaria gets inside your red cells. That's pretty disgusting, but that's what it is. Also babesia, if you know someone that had with Lyme disease, sometimes people get babesia, and it's like malaria. It crawls into your red cells. It's pretty gross. Okay, so let's just draw a couple of these things floating around inside and outside. So we can put viruses, and maybe some other kinds of miscellaneous bacteria. It says Yersinia likes to get in, so we could put maybe one of those long dotty things in there. There's some Yersinia. And some yeast cells or something. Well, this guy's really infected badly. And then let's do it right here in the bloodstream. Let's make a red cell. And put, this guy is malaria. And like I said, if you want to put babesia, B-A-B-E-S-I-A, babesia goes along with Lyme disease. Ticks carry it. So there's some babesia and malaria inside the red cells. And then we could put a worm egg floating by. Ooh, that would be gross. Nasty, nasty stuff. Okay, a little also bacteria floating in the blood there, not good. And this guy, Meningococcus, let's do some outside ones. The Meningococcus guy, the cocci, whatever, they're little round ones, and they form colonies. They stick. I read that they can stick to the inside of your veins and stuff, form little colonies. Isn't that gross? Meningococcus. And then it's hard for your body to get rid of them when they form a little colony like that. And then we could make another thing that's famous. We'll put a T cell floating around in the blood. It's a big T cell. And then we'll put some dots in here, and I'll put HIV. The virus that causes AIDS likes to get in your T cells. It's intracellular. And let's say I could put a little, let's pretend your Lyme disease hasn't got, there's Lyme, little spirochete. It's squiggly. It doesn't really look like a corkscrew. It kind of looks like a up and down little S shape like that. So there's Lyme floating in your blood. And then it gets out of the blood as fast as it can. It doesn't like the blood, but we can put another Lyme. We can put Lyme up here. So put a couple little pathogens outside. And then, let's see, strep is like a little chain. And staph is like, looks like a clump of grapes. So we'll just put all these little pathogens in the extracellular space. So some are outside and some are inside. And your body's going to have to decide where to look. Because remember that the T cell is going to get a little piece of pathogen, and it's going to see it on the macrophage, but it doesn't know where to go to look for it. It doesn't know where that piece came from. So, like I said, this is not a study of microbiology. It's just a little bit. It's worth a mention on our travels through, though, because over here, we're going to see how the T cells decide what to do about it. So we're going to make a cartoon macrophage right here. And he needs an arm reaching out over here, okay? So here's our little, mine looks really strange, see? He's looking over here. So this is cartoon. We're going to put a big mouth on him because he's the big eater, right? So that's how we know he's a macrophage. And he's going to have this little wet MHC2 clip coming over here with a piece of antigen that he's processed. And then over here we're going to have, I'm going to draw the receptor first so I get it right. Here's the receptor. So here we've got a T cell. Here's the naive T cell. He's not going to be naive in a moment. So it's going to match up. Or he's looking over here. What's going on? All right, matches up properly, and he's a T helper. Naive. There, got him labeled correctly. Okay, so it has this little, and it's going to have, remember the CD8 or CD4 over here. CD4 is going to go and touch. Checks it out. The CD4 says, "Yep, this is a macrophage I'm hooked up to." And look at that. There's a little bit of pathogen. All right, we need to go deal with this pathogen. But how does it know where to go? He's like, well, should I look inside of cells, outside of cells? What am I going to do? The macrophage also, remember we said it's another job is to secrete cytokines. The macrophage is going to secrete chemical signals. Chemical signals are generally called cytokines or cytokines. It's going to give off these chemicals, which there'll be little receptors here on the side to take them in. And it's a secret code. We don't know exactly, they're still researching this. We don't know exactly what goes on, but these chemicals will tell this helper, this CD4 helper, what to do, what to turn into. So when he gets activated, he can become, we're going to have to kind of draw one here and one here, one guy here, or this is something else he can do. He can turn into two kinds of helpers. Okay, what do you need help with? So he can turn into now we're going to draw a nice, a lively face, because big smile. He knows he's got a job now. Yay! I'm not naive anymore. He's no longer naive. He knows he's needed. And he can be number 1. We're going to put a 1 on his shirt and a 2 on his shirt. Nice big smile. I'm needed. Okay. So the macrophage will tell him what to turn into. If he turns into number 1, he's going to go look inside the cells. Go look inside. And he will go to alert other T cells. He'll say, look inside cells. So he's going to go alert the T cells. Because if it's hiding inside, remember, if it's hiding inside, the CD8 can kill it. Okay? So if it is not inside, if it thinks it's outside, it becomes number 2. And this guy goes, let's go look outside the cell. In which case, he will go to alert B cells. Now you want to look outside. B cells make antibodies, and antibodies stay outside cells. So the best plan of action, if you're looking for something outside the cells, go to the B cells and tell them to start cranking out those antibodies. Flood the system with the antibodies, and they'll float around and stick, because we know we're looking for something outside the cells. Antibodies aren't going to do any good here. They can't get inside the cells. But in this case, if the pathogen is outside, like we saw that worm that was covered with IgEs and the eosinophils are eating at it, the plan of action for if he turns into number 2 is to go alert the B cells. And in the next drawing, we're going to put it all together and see how all the parts function together.